Hello, everyone. Dr. S here, bringing you our 12th lecture, which is Bioenergetics. The goal of this lesson is to set us up for the physics of metabolism, essentially. We're going to introduce to you the energy currencies of the cell and understand why they are so, and we will compare the energetics of anabolism and catabolism, building up and breaking down, and see how generally they're regulated and interfaced. Our molecule of the moment you have likely heard of before. It is ATP, adenosine triphosphate. It almost always exists as the magnesium salt, so MgATP2-, and it is a universal energy currency utilized within biology. The delta G of its hydrolysis or breakdown is negative 50.5 kilojoules per mole in physiological conditions. And that's quite a bit of energy released, hence why ATP is the so-called chemical battery that fuels a lot of difficult reactions in biology. The activation barrier to its hydrolysis, though, just on its own, is 200 kilojoules per mole. All right, so my questions for you are threefold. Number one, what are its hydrolysis products? You probably know this already, but think about it, Google it if you must. Number two, construct a rough reaction coordinate for its hydrolysis based off the thermodynamic data provided. For the purposes of this exercise, I want you to assume there are no intermediates, just a simple progression between reactant to product. And then lastly, use your biological intuition to decide if ATP is kinetically stable. What does that term mean? Well, you could think about it and probably tease it apart on your own, or you could Google it if you had never heard that expression before. That is totally your choice, whether you do it one way or the other. As always, think about it, and we will poll the class and see what we think in lecture. It is time to talk about the circle of life. We get our energy from food. High energy compounds enter us, like carbs, fats, proteins, and DNA, and we break it down for parts, and we get rid of the high entropy waste, such as CO2 molecules and water molecules. This process of taking in complex molecules and then burning them, breaking them down, what have you, for energy or parts, all of that is catabolism. Degradation releases energy. That's not all we do, though. In turn, we assimilate the broken down food into our own body as complex macromolecules, such as proteins, polysaccharides, lipids, nucleic acids. Look what's happening here. We're making bigger, more intricate molecules. These are going to have very low entropy compared to the products of catabolism, which have very high entropy. Going up an entropic hill has a price. We need to pay an energetic tax to allow for anabolism, which is a set of synthetic metabolic reactions. And where do we get that energy from? ATP, NADH, NADPH, FADH2, all of these are forms of chemical energy. And we get these molecules recharged through catabolic processes. These drive anabolic processes. It's very yin and yang. You can't create without destroying. Now, the food chain leads all the way down to the autotrophs, aka producers. These creatures perform a very special anabolic process called photosynthesis. This is why the sun is the ultimate source of life energy, because photosynthesis is the only process in biology that we know of that converts physical energy, which is light, into chemical energy. This is what allows the fixation of a very high entropy waste product, CO2, into metabolites that are complex and high in energy, like sugars. Without that, life has nothing to work with. I want to re-emphasize that the net flow of energy from catabolic processes to anabolic processes is the key to what makes life alive. And since biology is organized and precise, we don't just light the food on fire inside of us and expect it to all run like a steam engine. No, the body is smart and it methodically extracts different kinds of energy and packages them in safe, stable, labeled, specific containers. Let's take a tour of the three types of containers or energy currencies we will be bartering with. ATP is an energy currency we all know and love. This molecule serves as a mediator of phosphate for sure. It's a phosphoryl donor. And since the hydrolysis of the gamma phosphate is very thermodynamically favorable, very exergonic, biology has learned to break ATP apart to power the more difficult transformations, one with very positive delta Gs. So ATP, said another way, is a source of free energy, so to speak. All right? ATP is not the only player in this economy. We are organic entities. We're constantly breaking and making carbon-carbon bonds inside of us. So life created carbon carriers like acetyl-CoA. Acetyl-CoA is a huge molecule that carries two carbons in the form of an acetyl group, shown in blue. All of this, that's just the Uber driver, so to speak. Lastly, we have our electron carriers. These are used to extract and inject chemical energy, bonds, from metabolites via redox reactions. The nicotinamide adenine dinucleotide pair can barter pairs of electrons via hydride transfer. When we say hydride, what we mean is H minus. Basically a lone pair, two electrons at a time. When we metabolize food for energy, what we are doing essentially is extracting out the hydrides from the food molecules and slapping them onto and charging up NAD plus into NADH. We'll talk more about that later. Now, like the NAD plus NADH couple, flavin adenine dinucleotide is similar, except it can do two electron chemistry as well, like its cousin, but it can also do one electron chemistry, aka radical reactions. So it's a little bit more flexible. The key to all of these energy, carbon, and electron currencies is that they are kinetically stable. we can charge them in one part of the cell and discharge them in another without worrying it will self-destruct before it does its job. We will be talking about these energy currencies extensively for the rest of the course, so I'll be shining a spotlight on each of them. And first, for ATP, many students are burdened with this misconception that ATP is a good energy carrier because it's like a loaded rubber band. I've heard that there's this high energy bonds between the gamma and beta phosphates, and you release that energy in that taut rubber band bond upon breaking it with a hydrolysis reaction. And that's a gross oversimplification of what's going on. ATP has a lot of energy, quote unquote, because its hydrolysis products are very low energy by comparison. And there's three reasons for that. First, we do attenuate some charge repulsion of the triphosphate moiety upon hydrolysis. This is what we mean by that third phosphate being kind of on against its will. The ATP molecule has 4 negative charges on it, as is. That's a lot of negative charge density in a small proximity. When you cut that bond, you divide the negative charges equally between a phosphate ion and ADP. So you reduce that internal charge repulsion, which hence lowers the energy of the products. The second reason why the hydrolysis of ATP is so favorable is resonance stabilization. The ATP molecule as a reactant has 2 phosphoric anhydride bonds that I will highlight in yellow. This is a phosphoric anhydride, and this is another phosphoric anhydride. Phosphoric anhydrides have limited competing resonance, like so. When I say competing resonance, let's draw the resonance structures like this. I'll put 2 explicit lone pairs on this bridging oxygen in this phosphoric anhydride, and let's ask ourselves, how many kinds of resonance structures can we draw? Well, I'm really only seeing 2. If these lone pairs collapse down, that'll kick up this pi system and put a negative charge on that oxygen and a positive charge on the bridging oxygen, right? And we could do the other thing if we swing the lone pairs to the left. Okay, so there's 2 additional resonance forms that we can obtain. Look what happens though when you liberate inorganic phosphate. If I were to draw it like this, what are all the ways that I can distribute those charges? You can draw this resonance form, or this one, and you probably get the picture by now. And then the last one, we'll put the oxo over here. So one of the products, inorganic phosphate, has 4 resonance forms alone, plus whatever resonance is left over on the beta phosphate of the ADP group. So all of that extra resonance is what's going to further stabilize those charges associated with the phosphate moieties. The third reason is solvation. By breaking a big molecule in half, we increase the surface area exposed to water. And hydration is usually an exothermic and favorable process, especially when we're dealing with charged entities like phosphate or ADP. I'll say usually an exothermic. All right, and we can visualize it by just sort of drawing a circle around phosphate and ADP. All of this space was inaccessible to water until we hydrolyze it. And by hydrating the areas I've highlighted in yellow, that will further lower the energy of the product state. So how does biology use ATP to get away with the unfavorable reactions of anabolism and such? To understand that, let's consider the enzyme glutamine synthetase. This is an ATP-dependent enzyme that reversibly converts glutamate into glutamine. Its reaction delta G is about positive 15 kilojoules per mole, which means this is a quote unquote not spontaneous process. It is thermodynamically unfavorable. I want us to get a feel though for how unfavorable it is though. We can actually guesstimate this by calculating its equilibrium constant, which is the concentration of the products divided by, of course, the concentration of reactants. So glutamine over glutamates. And there's an equation we can use to plug in the reaction delta G and some thermodynamic constants so that we can calculate this product to reactant ratio. If you plug and chug the Boltzmann constant and room temperature in Kelvin and whatever and rearrange this equation, you will get a much simpler term to solve for KEQ, the equilibrium constant, which is KEQ equals 10 to the power of the negative of the delta G divided by 5.69 kilojoules per mole. In this example, if you do that, it works out to 0.0025 about. And remember, with these equilibrium expressions, I always like to remind myself that these are ratios. 0.0025 over 1 is the same number as 1 over 400. And remember, these numbers have chemical contexts. Specifically, it's the ratio of products to reactants. And at equilibrium, equilibrium will favor the formation of the reactants 400-fold compared to the product. So this enzyme, without any external help, is going to favor a system that is mostly glutamate. What happens if we couple this reaction to ATP hydrolysis? Let's form a high-energy phosphoryl intermediate that should spontaneously form the desired glutamine. in a two-step process. So we took a little bit of a detour here to add on this phosphate, but let's see how that changes the energetics of this transformation. Now, there are many estimates for what the delta G of ATP hydrolysis is, but let's assume it's negative 50.5 kilojoules per mole. To find the net delta G of this new pathway, all you need to do is tally that with the original delta G of glutamine synthase, which was about 15 positive kilojoules per mole. When you combine that with the negative 50-ish kilojoules a mole of ATP hydrolysis, now our coupled reaction has a completely different delta G, which is now negative 35.7 kilojoules per mole. If we calculate the new KEQ in these conditions, well, that would be KEQ equals 10 to the power of negative 35.7 kilojoules per mole. Of course, multiplied by negative 1. So it works out to be a positive. And we divide that by the constant, 5.69 kilojoules per mole. The units cancel out because the equilibrium constant is dimensionless. And it may not be clear because my handwriting isn't the greatest, but it's to the power of. And that's going to be a huge number. 1.8 times 10 to the 6. AKA, we get 1.8 times 10 to the 6 products, glutamine, for every one reactant, glutamates. We went from a situation where product formation was negligible to a situation where reactant formation became negligible. Just by adding this phosphorylation step. Thanks to ATP, at equilibrium, the ratio is going to increase by 9 orders of magnitude. This is how ATP makes unfavorable positive delta G reactions possible in biology. Now, how quickly do cells use up their ATP stores? You may suspect correctly that varying tissues will have varying energy demands. Cardiac myocytes, for example, heart muscle, has an intracellular ATP concentration of 7.4 millimolar approximately. And that's one of the highest concentrations of ATP in the body. And that makes sense because heart muscle is constantly pumping, and so we need to have it full of energy. Now let's compare this to the ocular lens. The ocular lens's job is to sit in your eye and get sunshine on its face all day long. Pretty sweet life, if you ask me. As one of the most quiescent tissues in our bodies, it has an ATP concentration of 2.3 millimolar on average, which is on the absolute low end of the spectrum. And so I think these two extremes serve to show that yes, more active tissues have more ATP, less active tissues have less ATP. But I don't know, if you ask me, the difference between 7 and 2 isn't mind-boggling. And I think that brings me to my next point really well. It's not just how much ATP that a cell has that dictates its activity, but it's the ratio of ATP to ADP and AMP. That is what's important and what describes its current energy states. And there's a kind of a confusing equation over here called the energy charge equation, where it's the concentration of ADP plus half the ADP divided by ATP, ADP, and AMP combined. Usually, and when we're just talking in common parlance, scientists will kind of just know that energy charge is the ratio of ATP to ADP and AMP. All right, it's not exactly the same thing, but it gets the same message across. And I want to tell you that usually in most cells, 90% of adenine nucleotides are in the ATP form. So that tells you that the default energy charge when things are running normal is going to be on the higher side because we'll have a higher denominator. Excuse me, a higher numerator. So what this tells us is that biology has preferred that we have a Goldilocks concentration of adenine nucleotides. We need enough ATP to keep metabolism running, but we actually don't want so much ATP that this ratio, the energy charge, is not responsive to changes, all right? Not so much. The energy charge is insensitive to change. What do I mean by that? It's kind of like the low gas light on our cars. That gauge needs to drop down slow enough that we have time to recognize we're running low on energy and get to the gas station. We don't want it to drop so fast that it's going to deplete in the middle of the freeway, nor do we want it to deplete so slow that we forget it exists by the time we're in trouble. Make no mistake though, even 10, 15, 20 millimolar ATP will not last very long in a cell if it is currently active. In fact, it just takes a few seconds of muscle contraction to just wipe out the ATP reserves. But biology has a workaround. Phosphocreatine is a common exercise supplement because the body uses this as a phosphate reserve or phosphate reservoir. Creatine can help out with ATP production by essentially transferring its own phosphate sacrificially to freshly generated ADP molecules via an enzyme called creatine kinase. So to restate that, phosphocreatine recharges ADP back to ATP via creatine kinase, CK. And there's a good amount of this phosphocreatine the body can work with. About 95% of it is localized in muscle tissue. And the concentration of phosphocreatine is 5 times the amount of ATP. So that's pretty useful. And on a practical note, if you wish to employ this knowledge to further your own gains in the gym, I came across this paper over here, which did a study of peak plasma creatine concentration after a megadose via a supplement, such as the one shown over here. And that seems to be around, I don't know, a little bit past the hour mark across multiple different forms of creatine. So if you want to maximize your creatine stores, in theory, if you have it, I don't know, I guess 90 minutes before your workout, that seems to be the most effective time to do so. You may recall from general biology that ATP is an accessible, captured form of chemical energy that was derived from carbon fuels, like sugars and fats. At the chemical scale, we take energy from compounds by oxidizing them. This manifests as either a loss of electrons or a loss of hydrides, usually the same thing, and or a gain of oxygen atoms. Let's use methane as a simple example of a fully reduced quote unquote food molecule. As we catabolize it, what we do is extract out these hydride units, and we will use them to fuel ATP production. To remove those hydrides, the organism will replace them essentially with oxygen. And hence we go from something that is fully reduced like methane to something that is fully oxidized like CO2. So said another way, the more carbon-hydrogen bonds a molecule has, aka the more reduced it is, the more potential energy it contains. Likewise, fewer carbon-hydrogen bonds or more oxygens in that molecule will mean it has less energy. Let's apply this concept to the primary fuels used by biology, glucose and fatty acids. Right? Glucose is the preferred fuel. It contains only a modest amount of fuel potential, though. And the reason why is that it is already partially oxidized. Count them up. We have 1, 2, 3, 4, 5, 6 oxygens in that molecule. That depletes the amount of energy available by occupying spaces that could have been carbon-hydrogen bonds. Let's compare that to coconut fat, lauric acid, which was our second molecule of the moment. It is not preferred. Even though it is a great fuel source, and it is way less oxidized than glucose. I mean, look at all of these carbon-hydrogen bonds in that fatty tail. And if you look at the polar head, we only have 2 oxygens. So I have this question that I'm posing to you. What is the chemical logic of using glucose for energy over fatty acids? Even though the carbon hydrogen to oxygen ratio is much larger in lauric acid than it is in glucose. Pause the video and think about it for a minute. And once you do, you may realize that there are several differences between sugars and fats, one of them being solubility. We are aqueous organisms. Glucose dissolves better in water than lauric acid or any fat. And so it's much easier for the body or the cell to traffic glucose molecules throughout it for burning rather than lauric acid. And once it's time to chuck that food molecule into the furnace, we have to consider innate reactivity next. Each of those oxygens on the glucose molecule, yes, lowers its potential energy as a fuel. However, those oxygens are very convenient handles for chemical modifications. They're the in to cracking open that nut and releasing its energy. Lauric acid, by comparison, is a tougher nut to crack, even though it contains more energy inside of it. Now, I said in that last slide that we take energy from food molecules, from nutrients, by extracting out their electrons, and we use that to make ATP. Well, those electrons don't go into ATP directly. Instead, we use redox carriers like nicotinamide adenine dinucleotide, NAD+, to hold on to these electrons for safekeeping. And then later on, we will cache in these electrons to form ATP in a process called oxidative phosphorylation. We will cover that later. The important thing here to note is the way in which NAD+ picks up electrons. This nicotinamide group bears a redox center, which is quite unusual for organic compounds. In its oxidized state, the nicotinamide group exists as a positively charged pyridinium. It is aromatic, and therefore it will absorb very well in the UV region. And you can see that as the red curve in this electronic absorption spectrum of NAD+ versus its cousin, NADH. Now, I say that hydride and electron pair in terms of metabolism are more or less synonyms, and you can see that in the following case. Introducing a hydride is going to break the aromaticity of the nicotinamide ring, and we will add in a second H and a bond right over there, para to the pyridinium nitrogen. And we go from an aromatic system to a slightly less stable anti-aromatic system. As a result, it will be now absorptive in the near UV and less absorptive in the far UV. This NADH that we form is reduced. The NAD+ from which it came from is oxidized. There is more energy in the NADH molecule, and it's kind of like a wind-up toy. It will stay anti-aromatic if left untouched, but the tiniest trigger is all that it needs to revert back to NAD+ and release that energy stored in the form of hydride. It is therefore kinetically stable, high energy, but metastable. How does this NAD plus NADH couple work in serving its goal of extracting out electrons? Alcohol dehydrogenase is a good example that shows how this couple works in real life. Ethanol is actually extremely poisonous, but the enzyme alcohol dehydrogenase very quickly detoxifies ethanol into the less toxic acetaldehyde through a process called hydride abstraction. And briefly, how this reaction mechanism works is we start with NAD plus complexing an active zinc ion within the enzyme. So what we're seeing right now is metal ion catalysis. We are also seeing catalysis by approximation. Because bound to that same zinc cofactor is a water molecule that will be displaced by an alcohol. In this case, let's say it's ethanol. So what happens when we insert the ethanol into the zinc position? Well, we will lose the hydrogen on that alcohol hydroxyl through a process of acid-base catalysis. And that maximizes its ability to bind the zinc and approximate its correct position. That hydrogen is not what ends up on our NADH molecule. Instead, it is the carbon-hydrogen bonds vicinal to the hydroxyl group that will donate the hydride to the para position of our pyridinium moiety, like so. We will follow the arrow, like so, like so, like so. We will have an electronic rearrangement occur that ends up with NADH reduced as the product. Remember that we started with NAD plus oxidized. And this rearrangement results in the conversion of an alcohol to an aldehyde, which leaves and water will take its original position on the zinc ion again. This is a classic example of a mechanism that is substrate oxidative. Whenever we have a reaction that involves NAD plus being reduced into NADH, this is a great exemplar of how the enzyme achieves that transformation. This is catabolism in a nutshell. So the main job of NAD plus is to oxidize nutrients or poisons in ethanol's case and ferry those electrons towards ATP synthesis machinery. We'll talk more about this later, but NADPH is a very similar molecule to NADH, the main chemical difference being it has an extra phosphate group on the adenosine moiety. Functionally, though, we see NADPH perform substrate reduction or anabolism, whereas NAD plus is mainly seen in substrate oxidation or catabolism. The best example in this course of NADPH in action is fatty acid synthesis, which we'll discuss in a little bit. But this is a subtle point, all right? The NADPH is going to serve primarily to donate hydrides to less complex molecules and facilitate their building up into more complex ones. If you see here in this excerpt of fatty acid synthesis, we start with an oxidized precursor compound, one with a carbonyl group in it, and then adding in two equivalents of NADPH converts this oxidized low energy group into a higher energy reduced methylene unit. And this is a subtle point, but you may wonder, well, how is biology this organized? How does it know that NADPH is reserved for reducing substrates and NADH is reserved for powering ATP synthesis machinery? And the answer is it comes down to this phosphate that I mentioned earlier. It's a metabolic tag, it's not decoration. It's going to tell our enzymes, hey, these electrons are reserved for building or reducing macromolecules. The absence of that phosphate means that the substrate can't bind reductive enzymes, and the ensuing NADH molecule will have nowhere to go except the mitochondria to help give us ATP. So I'll repeat that this is a binding tag recognized by anabolic enzymes. FAD has similar redox properties to NAD+. It's just a little bit more complicated. Its redox center, which is called a flavin group, is famously lemon yellow. You can see a picture of that there. It's really pretty. When it is oxidized. So this is a picture of that molecule in the oxidized state. And unique to flavin, we can do sequential one electron reductions, like so. Or we can do chemistry more similar to the NAD plus NADH couple and do a hydride transfer, which is two electron chemistry, and completely bypass this semiquinone intermediate and go straight from the oxidized flavin to the fully reduced flavin. And you can see that the bonds we're adding are shown in blue here in the case of the semiquinone radical, and there in the case of FADH2. I don't expect you to know the details of this chemistry, just know that sometimes biology needs to do radical chemistry, and this is how. And in this slide, I'll now describe how acetyl-CoA works. It is essentially an adenosine moiety conjugated with vitamin B5, also known as pantothenate or pantothenic acid. And this whole group combined together constitutes a coenzyme known as coenzyme A, CoA for short. And coenzyme A basically chaperones whatever it's attached to across different metabolic circuits in the cell. And of course, in the case of acetyl-CoA, its job is to relay the 2-carbon acetyl group across protein, carbohydrate, and lipid metabolic pathways. The way that coenzyme A can achieve this is through its thiol group at the business end of the pantothenate moiety. Thiols can readily form thioesters between the pantothenate and important metabolites such as acetyl groups, which is shown in blue over here. The thioester group is metastable or kinetically stable. Metastable or kinetically stable. And what that means, again, is you can form it, you can stick it in a bottle, you can stick it in a cell, but you can break it apart easily when it is necessary. If we were to compare thioesters to oxygen esters are going to be more stable due to the increased resonance and orbital overlap between these two oxygens, shown over there. That will reduce the delta G for ester hydrolysis. But if you compare that to thioesters, it's harder to get that kind of delocalization when you're dealing with a very large sulfur atom. And so this thioester is way more prone to hydrolysis or nucleophilic attack. And that's also why coenzyme A is a good 2-carbon carrier. It not only holds on to the acetyl group, but it activates it for the next enzymatic step it's destined to experience. The last thing I'll say about acetyl-CoA is, isn't it curious how these carriers, acetyl-CoA, NAD+, FAD, they're all adenine nucleotide derivatives. I wonder why that is. Pause and think about that for a second. It's not by accident. In this slide, I'm going to show some very, 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 very simplified metabolic maps. Metabolism is complex and it is intertwined. Biology, for example, makes and breaks amino acids, carbs, lipids, nucleotides, and even less standard compounds like xenobiotics and foreign drugs. Secondary metabolites, funny conjugates like glycans and whatnot. And it's going to do all of these on the same metabolic landscape. As we spend the rest of the course teasing apart these pathways, you'll notice emerging patterns. You'll see certain metabolites like acetyl-CoA moonlighting in many, many pathways. You'll see the pathways come together and converge and then split apart or diverge, which means that a lot of these pathways depend upon each other. It's all interconnected. And that's what makes biochemistry, in my opinion, so maddeningly interesting. If you change one cog here, the effects of that mutation will trickle very far up and down the stream. I think I've mentioned it a few times now that cellular currencies like ATP, NADH, acetyl-CoA, and so on, once we make them, they are metastable or kinetically stable, meaning they will degrade over time because they are reactive. NADH and FADH will oxidize in air slowly. ATP will hydrolyze in water slowly. The only way we can cash them in on a sensible timescale is with enzymes. And biology is smart about that. If we made and used ATP at the same rate in equilibrium, no life would occur because we would get, again, a pointless loop reaction, a futile loop. So there's 3 methods biology uses to toggle between anabolism and catabolism, or currency making versus currency breaking. So what are those methods? The first one being monitor the energy charge of the cell. ATP will stimulate many anabolic enzymes. At the same token, ATP suppresses many catabolic enzymes. The reverse is true for AMP. AMP will stimulate catabolic enzymes, and AMP will suppress anabolic enzymes. And you can get a visual of that in this following plot, where we measure the relative rate of catabolism and anabolism as a function of energy charge, or the ratio of ATP to everything else. When we have a lot of energy charge, anabolism takes over. We have energy to spare. Let's use this bumper crop to build some stuff. When ATP is depleted, we need to activate our ATP generating pathways via catabolism. And that is where we will reign supreme when we are energy starved. Take a note here that anabolism and catabolism do occur simultaneously. They are just not to the same extent. They are not in equilibrium. That's the key takeaway. The cell will not be anabolic or catabolic. It will be mostly one and only slightly the other. The key here is you control the enzymes, you control the flow of energy. The second method of regulating anabolism and catabolism involves choke points. Many catabolic and anabolic pathways share common enzymes, but there's usually one or two steps that feature a unique branching or bypass enzyme that catalyzes an irreversible rate determining step. So, irreversible reactions. Whose specific enzymes can be activated or inhibited allosterically. Okay, what does that mean? I like to use this graphic to illustrate that point. In this process, we have a generic catabolic process and anabolic process, where the net flux of metabolites in the metabolic mode goes downward towards products. And in anabolic mode, we're moving in the reverse direction towards reactants, A. This could be, for example, glycolysis and gluconeogenesis, breaking sugar and making sugar from scratch. Enzyme 1, enzyme 2, enzyme 4, and enzyme 5 are identical in both pathways. These enzymes are reversible. Both pathways are identical, save for enzyme 3 and what is that? Enzyme 6? Enzyme 3 and enzyme 6. When the cell decides it needs to enter catabolic mode, instead of toggling every single enzyme in this pathway, it just needs to focus its efforts on regulating this one over here. It will turn that enzyme on and maybe this enzyme off. And that ensures we have a net flow of A to P in catabolism. Much easier than toggling every enzyme. Just focus the attention on the choke point. In anabolic mode, it's the reverse. Maybe this enzyme is irreversible in the upward direction. So let's activate this one to do anabolism and shut down that. we don't have to touch the common enzymes. They're still happy to run in either direction, but traffic will exclusively run towards the reactants when we only turn on that enzyme in that irreversible choke point. The third method is called compartmentalization, aka spatial control. Anabolism and catabolism may happen at the same time, but not in the same place. For example, in plants, sugar is made where? In the chloroplasts. Where is sugar burned? In a totally different organelle. In technically the cytosol is where it begins, but it ends up in the mitochondria. Both are different than the chloroplast. This is a way that we can ensure both will happen simultaneously, but not together in a feudal loop. The last strategy is genetic control, also known as temporal control. On top of all of the methods I described beforehand, cells can change the levels of anabolic and catabolic enzymes over time in response to certain hormones and signalers. Can adjust enzyme transcription via signalers. And we will see all of these four methods in action as we delve deeper into carbohydrate and lipid metabolism. So buckle in.